welcome back. In this video, we are going to continue with the eye, and we are going to be learning the pathway of vision and go in a little bit more details in some of the structures we've already learned. So what I want you to do is sit here and look at the picture and pause it. And what I want you to do is imagine you were a beam of light and name all the structures you have to go through, plus every time you hit a fluid. So structure and fluid, structure and fluid, structure and fluid. You should do that 3 times, okay? And stop with the word retina. So go ahead and stop it. Okay, so if you did that, including retina, you should have had, I think, 6 steps along your way. And remember, you are only naming structures that the light beam is actually passing through, not things on the side. Okay, so is light going through the iris or is light going through the pupil? You can't have both, so make the wise choice. All right. So hopefully what you did was you did cornea, anterior chamber filled with aqueous humor through the pupil, posterior chamber filled with aqueous humor, lens, vitreous chamber filled with vitreous humor, then retina. Okay. So we stopped on that word retina because before it was just this beautiful yellow line, but in reality, here's what the retina looks like. So when we're done learning about the retina, come back to this picture and see if you can name all the things we're going to learn about. Okay, so now, I hope you've noticed I flipped the direction of the eyeball. And that's because students get used to looking at the eyeball one direction only, and if on a quiz or a test it's the other direction, then they're confused because they know the thing's going this way and they can't make them going the other way. So in this case now the anterior part is to the right. So can you still see the cornea, the anterior chamber, the pupil, the posterior chamber, the lens, the vitreous chamber? Because I can see all those structures on that real picture on the right. So hopefully you can too. So now we are back here where the retina is, and this right over here is the retina. So the light is coming in on this one from the right side, and what I want you to realize, there are going to be 3 layers here of neurons. There's this pink layer of neurons, there's this lilac color of neurons, and then there's this whitish area. Okay. Now, yes, there are these bluish purple cells in between that we're not going to learn about. And yes, when you look at these whitish ones, when you look at the other ends of them, they're labeled rods and cones because some of them have a cone heads and some of them are much taller but skinnier. So yes, those are your rods and cones, your photoreceptors, that 3rd layer of neurons. And what I want you to realize is that the top part of all the rods and cones basically goes into the last layer, the outermost layer of the retina, which is a layer of pigmented cells. Okay, so this layer of pigmented cells is between all the neurons on this side and the choroid on the other side. Or if you flip the eyeball going the other direction, the light was coming this way, you would have neurons, 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 pigmented layer, then choroid over there. So the whole point of these pigmented cells is it keeps light from continuing to go. It's going to be stopping at this area. So with rods, these are the photoreceptors that are going to be activated in low light. Basically, they see shades of gray. So yes, you can call it black, but it's really dark, 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 dark gray. And yes, you can call it white, but it's really dark, 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 dark gray. Okay, so just think the whole spectrum of gray shades. Okay. And rods are mostly out in the periphery of your retina. They're not going to be anywhere close to your macula lutea. Okay, and each eyeball has more than 100 million of them, so there are lots of them. Cones, on the other hand, they require more light. You have to turn the lights on to activate these, and we're going to have cones. Most people have cones in 3 colors, blue, red, and green. Please notice these are not the 3 primary colors, because the 3 primary colors are blue, red, and yellow. There are no yellow cones, they are green cones, and I'll explain what I mean by blue, red, and green later on. These are much more towards the center of the retina, and as opposed to more than 100 million rods, we have less than 10 million of these. Okay? So we have less than 10% cones than we do rods. What is that orange arrow pointing to? Okay. So if you were to go to the right side and you were to have light come straight back right through the middle of the pupil, do you see how it would be hitting right there? So what structure is going to be right there? What is that little tiny little depression you can see right there? That's right. That's the fovea centralis. Okay. So let's look at all these retinal neurons. And on the left, I have the cartoon drawing, and I'll get to in a minute. So let's start on the right with the photomicrograph, because I know if I don't go through that, you all are just going to ignore it. So this is the surface down here at the bottom. This is the deepest part, and this is the outermost part. So right in here at the bottom of your picture is where all the vitreous humor would be. And so we are going to have a layer of neurons here, a second layer of neurons there, other cells we don't talk about, our third layer in here. And then you see this dark layer? That dark layer, that's your pigmented layer of your retina. And then out here is the very vascular. Do you see all the blood vessels? The very vascular choroid, because the choroid has to supply all the nutrients to the retina. The retina doesn't have any blood vessels. So if you've ever heard of somebody having, especially diabetics, getting retinal detachment where the retina pulls off the choroid, that is an eye emergency, and they go in with little lasers and they to retack it back down to the choroid. So yes, you're going to kill all the little photoreceptors at the little places where you're basically frying it back into place, but otherwise you would lose all your vision, the whole big part that detached. Okay, so now let's look at the cartoon area. So 3 layers of neural tissue. What has to happen is the light has to, in humans, has to go all the way through to find the first layer we're interested in, which are our rods and cones, which are these purple and yellow cells. They're going to generate an action potential, which is then going to be relayed through neurotransmitters to these pink cells, which I hope you recognized as bipolar neurons, because they are. All right, so where else have we had bipolar neurons? Yeah, some other special senses. All right. And then these bipolar neurons will release neurotransmitters, which will then interact with this third layer of cells known as ganglion cells. So yes, technically, I guess your entire retina could be considered a ganglion, because it's all these nervous cells, cell bodies of neurons outside the central nervous system. So yeah, the whole thing could be called a ganglion. We just call these ganglion cells. And then look what happens is really kind of cool. All the axons of all the ganglion cells are now the area that's closest to the vitreous humor. And so they all travel together, and they're all going to merge, and they go out through that optic disc to form the optic nerve. Okay. So in all the mammals, the cells are arranged this way. The rods and cones are the deepest, bipolar neurons in the middle, ganglion cells are the more superficial. But in other animals, whether aquatic or otherwise, no, I said that wrong. In humans, that's the way it's arranged. But in other animals, they're arranged the opposite direction. So when light comes in, the first layer it's hitting are the rods and cones, which then synapse the bipolar neurons, which then synapse the ganglion cells, which then go out the back end. But humans, we do it this way. Don't ask me why. I have no clue. Because it gives us a blind spot. All right. So light must get all the way to the deepest of the 3 layers of neural tissue in order to activate the photoreceptors, but light is prevented from going into the choroid or sclera or into the back of your brain or wherever you think it might be going because of that pigmented layer. And if that retina detaches from the choroid, you will lose your vision. So let's talk about these photoreceptors. When we look at these photoreceptors, there are multiple parts. There's like this cell body part, and then there's what's called an inner segment and outer segment. Okay, the only thing we're going to talk about are the outer segments. So the outer segments are the parts that are closest to that pigmented layer. So the cone head of a cone, and then the part of the rod that's furthest away from the vitreous humor, okay? And these outer segments are actually embedded into that pigmented layer. If you look in the outer segments, I think you can kind of get the idea by looking at all these lines that there's layers of stuff. And basically what there are, there's hundreds of little flat discs. And the new discs start down at the base, closer to the cell body. and then they work their way closer to the pigmented layer, and it takes about 10 days for them to go all the way up. And once they hit the pigmented layer, there are cells in there that do phagocytosis that get rid of all the debris. Okay. So in these flat discs, there are photopigments, okay? And there's a couple of different kind of photopigments, and they're different between rods and cones, so that's why I'm just using the word photopigment for right now. But all the photopigments contain two things. They contain a protein, which is known as an opsin. And each of those opsins has to do with a different wavelength of light, as we'll see shortly. And that protein is attached to a vitamin A derivative known as retinol. So I put a picture of carrots there to remind you, vitamin A. If you want night vision, eat your carrots, okay? And this vitamin A derivative, retinol, this is the molecule that absorbs light. Eat your carrots if you want to be able to absorb light, okay? So with light, what's going to happen, we're going to go in a whole lot of detail when you have the physiology of light. But with light, what happens is this photopigment is going to split. We're going to have this series of reactions. Ion channels are going to close, all kinds of things, and signals are going to go to neurons in our retina. All right. So as I said before, rods mostly in the periphery, many rods. And what the rods do, more than 100 million. So what the rods do is they do a lot of convergence, where many rods will go to a few bipolar cells, and then those few bipolar cells will go to one ganglion cell. And so basically each ganglion cell is bringing information from an entire square millimeter of the retina, which is quite a large area for one neuron to carry. In contrast to that, the cones are near the fovea centralis, and the fovea centralis is where we have our sharpest, clearest, most colorful vision. And near the fovea centralis, one cone goes to one bipolar cell, which then goes to one ganglion cell, which gives us a super sharp image. So only what's affecting one cone gives us that image. So instead of being a millimeter, it's 1/1000th of a millimeter or a micrometer whose information goes on to the ganglion cell. And as I said before, 3 cones, their pattern of being activated is going to give us all our various colors. And this is a little map showing the placement of rods and cones. So if you kind of like block out everything around it, I think you can see that you can see yellows and greens and reds towards the center of the eye, and all you see around the periphery is black. And then my last question for you, this area right here where you don't see any dots, what is that? What structure is that? Yeah, that's the optic disc. So remember, in the optic disc, all those axons of ganglion cells are exiting. Okay, so they're all going like this. So there's no rods and cones sitting in front of it. So that's your blind spot. Okay, now the blind spot is not straight back in the center. The blind spot is where the fovea centralis is, your sharpest vision. The blind spots on each eye are slightly lateral and down a little bit. Okay, and so that's why if you're looking with 2 eyes, you won't get 2 blind spots at the same point because they're not lined up in the same place. All right. So, should we do that pathway again? Yeah. So light hits I have to figure out my angles. Hits the cornea, enters the anterior chamber filled with aqueous humor, goes through the little pupil, enters the posterior chamber filled with aqueous humor, hits the lens, enters the big vitreous chamber filled with vitreous humor, and then before we used to say retina, but now we're going to change it because now light strikes the rods and cones. Action potential. That synapses with bipolar cells. That bipolar cells considered the primary neuron. That synapses with ganglion cell. That ganglion cell is considered the secondary neuron. It exits the eyeball through the optic disc, or the axons of all the ganglion cells exit the eyeball through the optic disc to form the optic nerve. Okay. So you have a close-up in the middle, and notice in the close-up in the middle, you can see all 3 layers of neurons there. So cute. And then you can see the little axons coming off in the big optic disc area, and you can see that there's no rods and cones in that area at all, because if there were rods and cones, how on earth could it possibly exit? And then you have a small picture at the top right showing the overview. On the left, this is the view that when doctors are, or your optometrists, when you go to get your eyes checked and they take that ophthalmoscope and they give you the drops to dilate your pupil and they shine that light in your eye, this is exactly what they're seeing. So when you look at this, do you see all these red structures? Okay, so these are all blood vessels, and you can see they're all coming from this area, which has this sharp, whitish light with a sharp edge to it that I'm circling around. So this is your optic disc area. And then when you go off to the side, do you see this darker area? That's your macula lutea over there. So that's in the center of the eyeball, and this one is off to the side. Okay. So now we're at the optic nerve. Okay. So before we continue on, let's talk about what happens, because we have two eyeballs, okay? And our eyeballs are both forward facing, as opposed to animals that have them on the side, like horses, which can only see one side or the other. They can't see it both. Because ours are in front, they both have visual fields like this, where they can see out at angles, and what you notice from the picture, like the left eye can view the blue area, the right eye can view the yellow area. So we have this part in the middle where the visual fields overlap, and that gives us what's called stereoscopic vision, which basically allows us to do depth perception. When you close your eyes and you try to figure out where something is exactly, it's a lot harder to measure depth than it is with two, and you can say, okay, I can clearly see that's 2 feet away. All right, so from the optic nerve, basically I've written out the rest, so there's no guessing, but from the optic nerve, we're going to have the optic chiasm, and look what happens in the optic chiasm. So let's stick on the blue side of the eye. So what these blue lines are representing, all those axons from the ganglion cells exiting, forming the optic nerve. And all the axons that are coming from the lateral part of the eyeball at the optic chiasm stay on the same side of the brain. But all the ones coming from the medial aspect of the eyeball will cross and go to the other side. And that happens for both of those eyes. So that's why the optic chiasm, the cross, that's why we have the cross, because half the fibers are crossing. So why is that good? Why is that bad? Because if you wipe out one optic tract, you haven't made yourself completely blind. It's not like losing one eyeball. Okay, it's like losing half of each eyeball. All right, so optic nerve, optic chiasm to optic tracts. Where do those optic tracts attach? Okay, so they're going to enter the brain, anterior to the pons, and where the majority of those axons are going to go to is to the thalamus, and they're going to synapse in the thalamus, because as we learned before, the thalamus is our major sensory relay center. And then from the thalamus, we would then pick up a tertiary neuron, which goes to the optic lobe, and when it goes to the visual cortex, you would be able to interpret it as, oh yeah, that's a red object that's 2.5 feet away from me. Now, notice I said most of them, okay? If you are visually tracking something, like playing ping pong, or tennis, or watching the Grand Prix, something like that, some neurons will then go to the superior colliculus, because that's the visual reflex traction center of the brain. And so you can see that by these little areas over there, okay? And then these other ones, these lower two ones, that's not meant to represent the inferior colliculus. That's meant to represent that there are a few specialized neurons that go to the midbrain, and those are for the ones, like for instance, for the pupil reflexes and for something called accommodation, which we will get into. So that's the end of your nerve impulse pathway. Here are a couple pictures to show you, so you clearly can see an optic nerve going to optic chiasm to optic tract, and you can see fibers diving in here to the thalamus, and then you can see fibers coming out. You also can see fibers coming out of the midbrain, and they're all going down here to the occipital lobe. Make it all the way to the cortex of the occipital lobe for you to be conscious of vision. All right, so what does the brain see? Everything is flipped, what the brain sees. What's on the right is on the left, what's on the left is on the right, and everything's upside down. So this is how babies are born. And then what happens is your brain mentally is flipping images all the time so that things are where you are. And the reason why we know this is because we can make specialized glasses which will flip everything right side up and give those to an adult, and for about a day or two, like you're bumping into everything and your brain hasn't switched things the way they are. So like if you're reaching for the soda, which is over here, you reach over there because your brain hasn't made that transition, and then you can go to sleep, and then you can wake up the third day and, oh, everything's worked out now. Okay. So that's kind of cool. All right. So now what I'd like you to do is I'd like you to discuss with a partner your pathways, everything we talked about. So there's the general view, there's your retina in more detail, and here are your last pathways. So go ahead and stop that, go over. So you don't even have to stop it because that's the last slide in this section. So I will see you again shortly, and thank you once again for working hard.